Hello dear students, welcome to this program. Today we are discussing an important topic, that is metabolic pathways. Before discussing in detail metabolic pathways, let's first know its objectives. Introduction to metabolic pathways, regulation of metabolic pathways, bioenergetics and metabolic disorders. Metabolism is the collection of biochemical reactions that occur within a cell. Most of these reactions can be grouped into metabolic pathways containing a sequence of chemical reactions. Each reaction is catalyzed by a specific enzyme, and the product of one reaction is the substrate for the next. The enzymes constituting a metabolic pathway are usually confined to a specific region of cell, such as the mitochondria or the cytosol. There are a very large number of metabolic pathways. In humans, the most important metabolic pathways are glycolysis, which involve glucose oxidation in order to obtain ATP. Citric acid cycle, also called as Krebs cycle, which involves acetyl-CoA oxidation in order to obtain GTP and valuable intermediates. Pentose phosphate pathway, which involves synthesis of pentoses and release of reducing power needed for anabolic reactions. Urea cycle, which involves disposal of ammonium ions in less toxic forms. Fatty acid beta oxidation, which involves fatty acid breakdown into acetyl-CoA. Gluconeogenesis, which involves glucose synthesis from smaller precursors. The compounds formed in each step along the pathway are called metabolic intermediates or metabolites that lead ultimately to formation of end product. The metabolic pathways of a cell are interconnected at various points so that a compound generated by one pathway may be shuttled in a number of directions. Metabolic pathways can be divided into two broad types. First, catabolic pathway, second, anabolic pathway. Catabolic pathways lead to the disassembly of complex molecules to form simpler products, while anabolic pathways lead to the synthesis of more complex compounds from simpler starting materials. Some pathways can function both in catabolic and an anabolic context. An example is the citric acid cycle, which not only breaks down acetyl-CoA but also participates in synthesis of amino acids. Such pathways are sometimes referred to as amphibolic. Now let's talk about regulation of metabolic pathways. In order to function properly, anabolism and catabolism in the body must be precisely coordinated. Metabolic networks sense and respond to information on the status of their component pathways. The information is received and metabolism is controlled in the following ways. First, allosteric interactions. Allosteric proteins contain distinct regulatory sites and multiple functional sites. For example, oxygen-carrying protein hemoglobin. The binding of molecules at sites distinct from active site triggers conformational chains that are transmitted to active site. Proteins displaying allosteric control are thus information transducers. Their activity can be modified in response to signal molecules or to information shared among active sites. Second, covalent modifications. Some regulatory enzymes are controlled by covalent modification in addition to allosteric interactions. For example, the catalytic activity of glycogen phosphorylase is enhanced by phosphorylation, whereas that of glycogen synthase is diminished. Third, hormonal control. The rate of synthesis and degradation of many regulatory enzymes are altered by hormones. This in turn helps in regulation of metabolism. Fourth, compartmentation. The metabolic patterns of eukaryotic cells are also affected by presence of compartments. The fates of certain molecules depends on whether they are in cytosol or in mitochondria. For example, fatty acids are transported into mitochondria for degradation only when energy is required, whereas fatty acids in cytosol are esterified or exported. Now let's talk about bioenergetics. Bioenergetics is the quantitative study of energy transductions that occur in living cells. Many quantitative observations on interconversion of different forms of energy led to formulation of two fundamental laws of thermodynamics, which are the first and the second law of thermodynamics. The first law is the principle of conservation of energy, which states that for physical or chemical change, the total amount of energy in the universe remains constant. While the second law of thermodynamics states that in all natural processes, the entropy of universe increases. Entropy is the degree of randomness or disorder of a system and is expressed as S. Any change in randomness of the system is expressed as entropy change, that is delta S, which by convention has a positive value when randomness increases. J. Willard Gibbs, who developed the theory of energy change during chemical reactions, showed that the free energy content, that is G, of any closed system can be defined in terms of three quantities, which are the enthalpy, that is H reflecting the number and kinds of bonds, second entropy, that is S, and the absolute temperature T in degree Kelvin. Free energy is expressed by the equation delta G is equivalent to delta H minus T del S. When a chemical reaction occurs at constant temperature, the free energy change is determined by enthalpy change, that is delta H, and entropy change, that is delta S. Delta H reflects the kinds and number of chemical bonds and non-covalent interactions, while the entropy change, that is delta S, describes the change in the system's randomness. A process tends to occur spontaneously only if delta G is negative. Yet, cell function depends largely on molecules such as proteins and nucleic acids, for which the free energy of formation is positive. To carry out these thermodynamically unfavorable energy requiring, that is endergonic reactions, cells couple them to other reactions that liberate free energy, that is exergonic reactions, so that the overall process is exergonic. The usual source of free energy in coupled biological reactions is the energy released by hydrolysis of phosphoanhydride bonds, such as those in adenosine triphosphate. At last, now let's talk about metabolic disorders. A metabolic disorder can happen when abnormal chemical reactions in body alter the normal metabolic process. Some of the possible symptoms that occur with metabolic disorders are lethargy, weight loss, jaundice, seizures, etc. However, the symptoms expressed would vary with the type of metabolic disorder. There are 4 categories of symptoms, which are acute symptoms, late onset acute symptoms, progressive general symptoms, and permanent symptoms. Now let's talk about types of metabolic disorders. The principal classes of metabolic disorders include acid-base imbalance, metabolic brain diseases, glucose metabolism disorders, hyperlipidemia, iron metabolism disorders, lipid metabolism disorders, and metabolic syndrome X. Now let's talk about inborn errors of metabolism. Inborn errors of metabolism can be divided into three pathophysiological diagnostic groups. First, disorders that disrupt the synthesis or catabolism of complex molecules with symptoms that are permanent, progressive, and not related to food intake. These include lysosomal disorders, peroxisomal disorders, and disorders of intracellular transport and processing. Second, disorders that lead to an acute or progressive accumulation of toxic compounds as a result of metabolic block. These include disorders of amino acid metabolism like phenylketonuria and maple syrup urine disease. Galactosemia, which is linked to sugar intolerance, also falls in this category. Third, disorders with symptoms due to deficiency of energy production or utilization within liver, myocardium, muscle, or brain. These include congenital fatty acid oxidation defects, gluconeogenesis defects, and mitochondrial respiratory chain disorders. This is all about the topic. Hope you have understood. Thank you.